Good afternoon, everyone, and welcome to the TMS meeting today. We have the pleasure to host Professor Francesco Angelucci. He is Professor of Biochemistry at the University of L'Aquila, and he got his master's degree in chemistry and the PhD degree on structural biology and biochemistry at University Sapienza in Rome, where he has been also postdoctoral fellow. He is an expert in drug discovery, and he got several honors. He has been invited as speakers at international meetings. He also obtained the Bioeconomy International Prize for Innovational Research in the field of biochemical research. Very importantly for Francesco is that he is supported by grants from the National Institute of Health in the United States, which are very prestigious grants, and also from the Italian PON grants. He has published several papers, over 65 publications I counted, and they are all around the drug discovery area. And today he will talk about the structural biology and drug discovery. Francesco, the floor is yours. You can share the screen and start your presentation. Thank you.
Okay. Good afternoon. Thank you, Anna, for your kind presentation, for your invitation. So I'm going to share the screen. So the title of my presentation is Structural Biology and its Role in Drug Discovery. This is a short outline of the seminar. In the first part, I will describe structural biology and the main experimental methods used, which are X-ray crystallography and cryogenic electron microscopy, while in the second part, I will show you the structure-based drug design study on a vital selenoprotein from a human parasite, which is the project that we are trying to carry on here in L'Aquila.
Francesco, we don't see the screen.
Wait,
Your
sorry.
screen? So
because I didn't share the screen this way.
Oh, okay. Okay, thank you.
Okay. No. Okay, so this is a short outline of the seminar, sorry. In the first part, I will describe the structural biology and the main experimental methods used, which are X-ray crystallography and cryogenic electron microscopy, while in the second part, I will describe the structure-based drug design study on a vital selenoprotein from a human parasite, which is the project we are trying to carry on here in L'Aquila. So let's go with the first part. And I don't know how to remove this one. Okay, doesn't matter. Anyway, so why is important structural biology? The first thing is to understand function, because if you know the 3D shape of a protein, or in general of a macromolecule, you can infer on the reaction mechanism, on its ligand binding properties, and on the physiological and pathological role. Second, if you know the 3D shape of the functional sites, or in general of allosteric site of a protein target, you can design small molecules that fit in it, inactivating the enzyme, and so making a new drug. And this part is called structure-based drug design. To convince you how much is important structural biology, I take advantage on this I will say data bank that is called Protopedia. So let me show the Protopedia main page. This is Protopedia. It is the Wikipedia of proteins. So each protein has its own page. And this site is very useful both for teaching and for learning. For example, I can show you very quickly. If you type hemoglobin, there is one page related to hemoglobin. On the right, you have the 3D model that you can rotate and see different features of the structure. So there is a lot of text, reference, new insights. But the page that I would like to show you on Protopedia is this one. Just a second, sorry. That is called highest impact structures. So in this page, there are macromolecular structures that change our views to understand several biological processes. I would say fundamental biological process. So you can see in 1953 the structure of the DNA aldehyde helix, and this was fundamental to understand at the molecular level the replication process. The first structure of a protein, myoglobin, the first structure of a lysozyme in 1965, and in 2000, the structure of the ribosome. And this structure surprised almost everyone because it showed that the peptide bond formation is carried out by a ribozyme rather than a protein enzyme. And if you scroll the page, there is another paragraph that is called structures saving the most lives. And you can see at the end, there is the structure of the HIV protease. And so let's back again to the presentation. And so understanding function, ribosome at work. This is a very short video in which several X-ray crystallographic structures are placed one after the other, and each crystallographic structure is in different conformational states with a different ligand bound. And you will see that when I will start the video, there will be the tRNA transfer with bound an amino acid brought by the elongation factor EF2. This is a prokaryotic ribosome, so the major subunit in orange and in brown, the minor subunit is in cyan and in blue, and the messenger RNA is this one in gray. So there is the RNA transfer with the nascent polypeptide in green in the P site, and in the yellow one is the tRNA, the RNA transfer in the exit position. So each step of this video is accompanied by a song, and each song in some way highlights the process that is going on. I don't know if you can hear the song, but this is the RNA transfer with EF2. And so it's trying to find the right codon on messenger RNA. When it finds the right codon, there are several conformational states that work as a quality control that end with the hydrolysis of GTP. And now you are seeing the conformational states that say that the codon-anticodon interaction is the right one. That is the GTP, and the yellow one is the phosphate that will release the site. Once the GTP is hydrolyzed, EF2 can be released by the ribosome, and the peptide bond can be formed. This film has been made by the Stites group, which is the person that solved the first structure of a ribosome and won the Nobel Prize. So this video is to show you the importance of structural biology to understand function. And the other thing is the structure-based drug design. So how much important structural biology to design drugs? And structure-based drug design is a cyclic iterative procedure that takes advantage of the 3D structure of a validated protein that target to design new chemotypes with high potency and selectivity, and so on new drugs. This is the procedure. It starts from target identification and validation. What does it mean, this first step? It means that you have to know that your protein is important, is vital for that cancer cell, that pathogen. And when you know this crucial biological information, You have to clone and express your protein. You have to solve its three-dimensional structure by X-ray crystallography or cryogenic electron microscopy. You have to run modeling and design by computer. You need to synthesize the compounds and evaluate against the protein target. And you can repeat several times this cycle after you reach a compound, hopefully with nanomolar affinity. And so you have a lead compound. And if you are lucky, you can go in preclinical trials. The first major success of the structure-based drug design approach are the HIV protease inhibitors against AIDS. HIV protease is crucial for the viral life cycle and proved to be druggable, as this video would like to show you. So you will see the protease that cuts the polypeptide generated by viral DNA into functional proteins that are necessary for viral progeny. Saquinavir was one of the first approved drug in 1994, and it binds in the catalytic cleft of the enzyme, blocking its function and working as a competitive inhibitor. The first procedure based on a structure-based drug design approach was used against COVID-19. And here you can see Paxlovid is a drug on the market made by Pfizer, is a very effective in inhibiting the main protease from COVID-19. What are the main methods in structural biology? So what are the methods that are useful to do these kind of things I show you in the previous slides? They are mainly X-ray crystallography and cryogenic electron microscopy. Both these methods have disadvantages and advantages. For example, to use X-ray crystallography, you need crystals of your target protein. And the problem is that not all the proteins crystallize. For example, membrane proteins are very difficult to crystallize. The advantage is that there is no molecular weight limits. I can solve the structure of a ribosome, I can solve the structure of an entire virus. And it is the method of choice for drug design studies because it can reach very high resolution. On the other hand, cryogenic electron microscopy, cryo-EM, has the disadvantage that it is not able to work with very small proteins, so with molecular weight less than 50 kilodalton. But it works in solution. It is very good for membrane proteins, and it is now starting to be used in drug design studies due to the resolution revolution. What is the resolution revolution in cryo-EM? So before 2013, the resolution reached by cryo-EM was about around 10 angstrom. And now cryo-EM can reach very easily near atomic resolution. And so you can see the orientation of the amino acidic side chains. And this is a very important feature if you would like to undertake a structure-based drug design study on the protein target you solved. What we need to solve the structure by these two techniques, we need pure proteins in high quantity, very expensive facilities to get the high resolution data, synchrotron light source for X-ray crystallography, and very powerful electron microscopes, and high performance computers, but this mainly for cryo-EM. So this is a short video. If you are interested, I can show you all the video after the seminar. But in this video tries to show to you the procedure in X-ray crystallography. It starts from preformed crystals that are here in this little drop and arrive to the solved structure. And so you will see this person that will pick up the crystals from this drop under a microscope, and then freeze them, and then ship them to the synchrotron light source. So the crystals are red because they are hemoglobin crystals. This is the crystal. This is liquid nitrogen.
This is a protein crystal. It's pretty small at just 1/10. And millions of protein molecules stacked inside it. Small.
And this is a synchrotron light source. This is close to Oxford in UK. You can walk in there.
Is used by scientists in many different ways, but are structural biologists doing X-ray crystal. This is a technique that's to look at the structure. The secret of success is growing crystals, which form together. Scatters X-rays. Resulting diffraction pattern, detected as in spots, is dependent on the internal structure of the crystal. Measuring the intensities and positions of these spots, we can work back to the structure of the molecule it is made from. It's a method that has opened our eyes to life at the molecular level.
Okay, so I stop the video and I would like to show you also this other video about the cryo-EM procedure. In this case, the video starts from purified protein in solution, and then you will see that the protein will be placed on a grid for electron microscope and put under the microscope, and you will collect several thousands of images, so bidimensional photographs of your protein in different orientation in solution, and then a computer try to build the three-dimensional model starting for the bidimensional information present in the micrographs in the images.
Microscopy starts with the preparation of a pure sample. The sample is frozen in liquid ethane so rapidly that ice crystals cannot.
Also here, the sample is freezed. Then I will explain why if you are interested.
Detected by a camera to produce an sample, different orientation of computer. Images are software is used.
This is the final 3D model.
To produce a high-resolution 3D structure. This is gamma secretase, a protein important in Alzheimer's disease. The structure of gamma secretase was determined by scientists at the LMB using cryo-electron microscopy. To visualize a protein at this level of detail allows us to better understand how the protein functions. It opens up the possibility to design drugs that precisely fit proteins and offers an exciting opportunity to treat diseases.
Okay, so I hope that I give you a general idea about structural biology and what are the main techniques used. So, second part, our project here in L'Aquila. These are the people involved in this project. I would like just to mention the people working in L'Aquila, in particular Rodolfo Ippoliti, that helped me to set up a structural biology unit in L'Aquila. Matteo Ardini is the key person for cryo-EM studies. Federica Gabriele, Francesca Fate, Lera Silvestre, and Marta Palermo did many of the X-ray crystallography experiments that I will present. So pterodoxin reductase is the focus of this presentation, and together with glutathione reductase maintain the redox homeostasis in almost all the organisms. They regulate DNA synthesis, defense against oxidative stress, and cell signaling. And for this reason, they are considered very crucial drug target in a plethora of human diseases. They pick up electrons from the DPH and they transfer to pterodoxin. In the case of pterodoxin reductase, pterodoxin is a small molecular weight protein, while in the case of glutathione reductase, GR, This enzyme takes the electron from the ADPH and transfers to the oxidized glutathione. Mammalian thyrodoxine reductase are selenoprotein. So they contain a selenocysteine at their C-terminus. They are homodimer, and here you can see one subunit colored in gray and the other in cyan, and in each subunit there is a FAD cofactor, a flavin cofactor. On one side of the cofactor there is a tyrosine residue with its ring perpendicular to those of the FAD, and on the other side there is a couple of cysteine. So you can see the same architecture here with the same color code. So tyrosine perpendicular to the FAD in the closed position, and on the other side the cysteine and the C-terminal selenocysteine. Once an ADPH enters the site in order to reduce the enzyme, this is the first step of the enzymatic reaction, tyrosine must swing out in order to make room to the ADPH that now can reduce the FAD and a couple of cysteine. Once the enzyme is reduced, electrons can be passed to the thyrodoxine substrate. This is a very short video that shows you the movement of the tyrosine and the entry of an ADPH. The problem, no, it's not the problem, sorry. Thyrodoxine reductase are very important drug target, are potential drug target in cancer, inflammatory disorder, and several infectious diseases. For instance, oronophin, a gold-containing compound and an FDA-approved drug, is used now. I mean, you can find it in the pharmacy. Is used to cure rheumatoid arthritis in humans and is now in clinical trials against leukemia and ovarian cancer. The problem is that thyrodoxine reductase are very difficult to inhibit in a selective manner and to approach by a structure-based drug design, mostly due to the fact that they contain a selenocysteine. In fact, selenoproteins are very difficult to obtain in high yields with 100% selenocysteine incorporation by heterologous expression. What is selenocysteine? Selenocysteine is the 21st amino acid. It is a very reactive residue. It is 100 times more nucleophilic than cysteine at physiological pH. And to be inserted in a polypeptide chain, it needs a selenocysteine insertion machinery. In particular, I don't want to explain all this complex machinery. There is the need of a particular RNA transfer that brings the selenocysteine on one side and that on the other side recognize a stop codon on the RNA messenger. So the problem arises when you try to translate this machinery in E. coli in order to carry out heterologous expression. Because in E. coli, there is a competition between this tRNA transfer and the releasing factor of the ribosome for the stop codon. And this results in about 50–70% of truncated protein and only 30–50% of wild-type protein. And this creates problem if you would like to carry out a comprehensive functional characterization of a selenoprotein. The other problem is that non-inhibitors of thyrodoxine reductase are promiscuous. They are mostly pains. Pains mean pan-SA interference compounds, and they are false hits, so false inhibitors, false selective inhibitors, very difficult to optimize. Mannic bases, curcumins, quinones, probably you know curcumins, quinones, catechols are all found as hits in the high-throughput screening against thyrodoxine reductase-like enzyme. But, as I told you, they are promiscuous because they behave in general as a general electrophiles. Thyrodoxine reductase are particularly sensible to pains as they are equipped with selenocysteine, which is the stronger aminoacidic nucleophile. In 2002, now I will try to make a story about our work. In 2002, a new thyrodoxine reductase-like enzyme was discovered by the group of David Williams in Chicago. And this is a particular fusion of a thyrodoxine reductase domain and two glutaredoxine domain as in its N-terminus. And this enzyme is able both to reduce thyrodoxine in the same way we have seen in the case of thyrodoxine reductase, but it is also able to reduce oxidized glutathione thanks to the additional GRX domains. In parasitic platyhelminths such as schistosome, this enzyme is the only one able or supplying reducing equivalents to the thyrodoxine and the glutathione pathways. And it has been demonstrated to be an essential drug target for the treatment of schistosomiasis, as after silencing of TGR expression, parasites die after few days. Oronofin, a non-inhibitor of thyrodoxine reductase, kills worms after few hours, and it has been demonstrated that TGR is the target of two old anti-schistosomal drugs used in the past to fight the disease, antimonium salts and Oltipras, but then dropped out due to the several side effects of these two molecules. What is schistosomiasis? Schistosomiasis is a neglected tropical disease. It afflicts about 200 million people in the tropical areas, and 200,000 die each year for disease complication. The parasite has a complex life cycle. It lives in the mesenteric veins in a human, and it produces about 1,000 eggs each day. Some of the eggs are trapped in the human body due to the granuloma formation, and this causes mental and physical impairment, hepatosplenomegaly, ascites, and in some cases, death. There is just one drug against the disease, that is praziquantel, that only in 2018 was administered to 90 million people. It is a very safe drug, but it has several important limitations. For example, it is not so effective against juvenile worms, I mean worms after 3-4 weeks post-infection, and less sensitive strains are emerging. So alternative to praziquantel must be sought, and TGR inhibitors can be a solution. And in fact, quantitative high-throughput screening against TGR, this is around 2008, several quantitative high-throughput screening have been carried out against TGR on more than 500,000 compounds at the NIH, and one compound, a furoxan analog, was discovered to behave better than praziquantel in animal models. And in those years, there was a lot of enthusiasm, as you can see from the several papers, commentary papers that appeared in Nature journal in that period. But furoxan proved to be metabolically unstable, and their further optimization was halted. In the same years, we solved the first structure of a TGR, in particular of the TGR from Schistosoma manzoni, but of the selenocysteine to cysteine mutant. We solved a very good resolution, and so we wanted to undertake a structure-based drug design study. We have the structure of the protein, we have the inhibitors from the high-throughput screening, and we try to solve the complex in order to understand the mechanism of action and to optimize these initial hits. But no matter how we tried, no structure was solved in that period. In 2017, things started to change, mainly for two reasons. In 2017, the group of Elias Zander and Karoliska solved the issue related to the incomplete incorporation of selenocysteine into thyrodoxine reductase. In fact, he used an UAG stop codon, which is recognized by releasing factor 1, and used an E. coli strain not expressing releasing factor 1. And this resulted in 100%, almost 100% selenocysteine incorporation, and this led us to fully characterize a selenoprotein, in our case, TGR for Schistosoma manzoni. And in the same years, also became clear that the fragment-based approach can be very good in finding new biologically relevant site to be exploitable in drug design studies. And so this approach can be very useful against difficult targets as pyridoxine reductases are. What is fragment-based approach by X-ray crystallography? Fragments are very low molecular weight compounds, very soluble, characterized with low affinity, but they are better than bigger compound and more effective compound to probe the chemical space in a functional site. There are several ways to optimize an initial fragments. The first strategy is called fragment growing. So you found a fragment in a pocket by X-ray crystallography or cryo-EM, and then you grow this initial fragment in order to increase the contact with the pocket. And so what you want to do is to increase the affinity of the resultant molecule for the protein target. The other strategy is fragment linking. This strategy starts from 2 or 3 fragments bound in the same site, and then you try to join these initial fragments by different chemotypes in order to increase the complementarity and increase the affinity of the resulting molecule for that protein site. And so we took TGR crystals, we selected more than 100 fragments from the high-throughput screening, and we tried to solve the structure of their complex. We collected more than 300 data collection at the synchrotron on these crystals, and we solved the relative structure. We found 10 different hits, 10 different fragments bound in a pocket very close to the NADPH binding site. So this is the pocket that we call doorstop pocket. It is separated from the NADPH binding site by the rotating tyrosine that in TGR is the tyrosine 296. You can see the same architecture here schematized. And so, why this pocket is the doorstop pocket? Because fragments here are schematized by this green rectangular shape binds to a pocket close to the rotating tyrosine, and they impede the movement of the tyrosine necessary for the NADPH entry and enzyme reduction. Our collaborators in medicinal chemistry, mostly using fragment linking, synthesized more than 150 compounds. These compounds were tested against recombinant TGR enzyme, recombinant human pterodotoxin, and they show very high selectivity. Some of them show very high selectivity with respect to the parasite enzyme. The ratio between reduced glutathione and oxidized glutathione has been measured in worms, and this is an important indication of target engagement because TGR is the only enzyme able to reduce oxidized glutathione in the platyhelminths. The compounds were tested against several life cycle stages of schistosoma, and they were also tested for toxicity against mammalian cells. So to continue in the process of drug design, the one that I showed you before, in the iterative procedure of drug design, we try to solve the structure by X-ray crystallography of TGR with these new, more effective compounds. But again, we failed. And so we decided to switch to cryo-EM. Matteo Ardini solved the structure. He got beautiful map of TGR in complex with one of the inhibitor, demonstrating the feasibility of this methodology to study this protein family of importance in several human diseases. And he found that one of the compounds is actually bound to the doorstop pocket. And this was very important result because it's telling us that the drug design strategy used so far is successful. And so we tested one compound against schistosoma infection in animal models, in mouse model. And as you can see here, treatment with just a single dose of one of the compounds at 100 milligrams per kilogram resulted in about 80% of worm burden reduction. And this value outperforms the lead activity criteria indicated by World Health Organization. This is the images of the liver of an infected mouse by schistosoma. The white spots are the granulomas around the eggs, and this is the liver of an infected mouse cured by one of our compound. And the same compound was also tested against juvenile worms. I remember to you that the juvenile worms are less susceptible to praziquantel, and in this case, our compounds outperform praziquantel, which is the drug of choice against schistosomiasis. So in conclusion, a new secondary site of a thyrodoxine reductase-like enzyme has been identified, and a new inhibition mechanism is proposed for all the family of thyrodoxine reductase. New lead compounds for TGR have been discovered, displaying schistosomicidal activity against culture worms better than the thresholds set by WHO. Some novel TGR inhibitors demonstrate efficacy against juvenile worms, significantly outperforming praziquantel, and these findings open a new avenue for the design of non-covalent inhibitors for TGR for schistosomiasis treatment. Thank you for your attention.
Thank you, Francesco. Very inspiring, I think, especially for the young people that are attending this webinar. So I think that we can start the discussion. The audience can either raise the hand and unmute themselves, or if you are shy, you can just write your question in the chat, and I can read it for Francesco.
Yeah, sure. Sorry, but I can't return to the main page of WebEx. Let me see if I... Okay, it doesn't matter. And I can hear you very well.
Okay, thank you. So I can start with some naive questions. Actually, it's not my field of research, but I think that this field is growing very fast, and there are a lot of new drugs now around, and probably we will have more in the near future. My naive question is, can this drug or this type of drugs or this strategy be active also on other type of parasites, or even, for instance, the malaria plasmodium, or this type of organisms that affect people, especially in certain countries? Is there any hope for this, or you have a strategy to expand to other parasites?
Yeah, actually, Anna, we are expanding this strategy to other proteins. An idea was to expand this strategy against Plasmodium falciparum thyrodoxine reductase, which is differently to TGR and to the human thyrodoxine reductase, doesn't have the cysteine residue, the selenocysteine residue, but it conserves the same mechanism of reduction by an ADPH. So this is principle possible. What we are doing with the other people in L'Aquila is to translate this strategy to the human thyrodoxine enzyme, trying to cure some kind of cancer. So we are not experts of cancer. This is the first thing that I should say. But we know that several people, especially one of our collaborators, Karolinska, that his name is Elias Arner, the one guy that I told you, he found the right way to insert selenocysteine in the thyrodoxine reductase enzyme, is actively work against cancer using inhibitor against this enzyme. But he is trying to use another strategy. He's trying to covalently attack inhibitors to the selenocysteine. This is strategy. He published now a paper on Science Translational Medicine on one of these inhibitors. He made also a company about this kind of inhibitor, but I don't know now what is the status of his work now. But tyrosine reductase is a very important target, as I told you, against cancer, and we are trying to translate our strategy also to the human enzyme.
Mm.
And we are in a good point because with Matteo we solved the structure possibly of the human enzyme with one of the same inhibitor of the same series bound to the dot-stop pocket, and so we are working on that.
Okay, thank you. And you mentioned in your talk that you would like to explain us why you need to freeze
Yeah.
yeah. So can you explain what is the
Yeah,
point?
sure.
Yes.
Let me come back to the yeah. I don't want to restart the video, but in X-ray crystallography, it's important to freeze crystals before shipping to the synchrotron, because the synchrotron light source is very intense, very powerful. So if you don't freeze the crystals, if you put the crystals at ambient temperature under the synchrotron, probably the crystal explodes after 1 second. So you can't collect data on that crystal. Consider that for a complete data collection in general, for a general symmetry, you need about 200 images, 200 diffraction images. And so you want to keep your crystal safe during data collection. And this is why you need to freeze them and to put under a nitrogen gas during all the data collection. For cryo-EM is the same reason. The only difference is that cryo-EM works in solution, and so you have the protein on the grid, but you need to freeze the grid. And what you want to do is to freeze the orientation of the protein in the solution. So in this case, when you collect several images, you have the negative photographs of the several orientation that you can have of the protein in solution. And this is very important because if you have several orientation of the protein solution, you will probably have a better structure, a better three-dimensional structure of the protein. So it is important that you freeze the sample because you want to make, you want to stop the movement, okay? And also because there is also for cryo-EM a problem of radiation damage of the biological sample. This is the other reason.
Yeah, it makes sense, yes. So let's see if in the audience there is any question. So don't be shy, just mute yourself. We have students from UK. We have also senior people attending. So I expect that from these people some good question can come up. Yeah, maybe someone is writing in the chat, and yeah. So what is? I have a question.
Yeah.
How did you get your grants from NIH?
Allora, yeah.
This is interesting, I think.
Allora, what we got is, I should say that we failed 3 or 4 times before we got the grant. Now we are in the list for having a new grant. We are waiting for the final pay line from NIH at present. We finish our money to carry on this research, so it will be very helpful to get new grants. Anyway, we got these grants because I should say the work on schistosomiasis, I mean, not so much people work on this kind of parasites. And we are the only people that are doing structural biology against this kind of parasite. And this is the reason why we set up during years a very fruitful collaboration with people at the Rush University Medical Center and at the University of Illinois in Chicago. So we make this group trying to work in the same direction. This is why, I mean.
You must be very good for getting NIH grants. So very
Thank you.
congratulations. Yes. We have a question in the chat, but Eve, can you unmute yourself? I think that you can make your question.
Sorry, Anna, I can't display the entire, I don't know how to do that, but I can see the question.
In the chat. No,
Chat.
but I think I'm asking Eve to unmute herself because she can ask the question directly. Eve?
Hello, can you hear? My microphone's not the best.
Yes, we can hear you.
I found this talk really interesting. I
yeah.
studied a little bit at university, and I can't really remember much, but my question was if cryo-EM is used more often nowadays for more modern researchers. It's a newer technique, I believe. And yeah, if it's a more beneficial technique to use.
Yeah, it's a good question. So cryo-EM exists, if I remember well, from first year of the 90s. What the new things is that in the first year of 2010, new detectors were discovered, and this create the resolution revolution that I showed to you. So cryo-EM can reach similar resolution of X-ray crystallography in the last years. Consider that in 2017, the Nobel Prize, people that made possible the resolution revolution won the Nobel Prize in 2017. And yes, cryo-EM slowly, but is outperforming X-ray crystallography, mostly because it doesn't need crystals, and also because it is very good to solve the structure of membrane proteins. And the problem with X-ray crystallography is that membrane proteins, receptor, ion channels do not crystallize. And this is the problem why X-ray crystallography is not so good for membrane proteins. But in fact, if you see Nature of Science journal, you will see that probably there is one structure each month solved by cryo-EM of a membrane protein. And so I think that in the next years, cryo-EM will be the method of choice in structural biology.
Yeah, because I remember learning about how difficult it is to crystallize proteins, and that being like a major limiting factor for X-ray crystallography.
Yeah, is the limiting factor in X-ray crystallography. The problem is that to crystallize proteins and membrane proteins do not crystallize very well or at all.
Okay, thank you for your answer.
You're welcome.
Okay, thank you. So we have Flavia also with a question. Flavia, do you want to unmute yourself?
Yeah, thank you very much for the talk. It was extremely interesting. I was more curious about understanding whether your findings could be also transposable to central nervous system diseases.
You mean cancer disease or neurodegenerative diseases?
The neurodegenerative diseases.
I don't have idea about this. Sorry. I don't know if target-ligand reductase is an actual target against Alzheimer or Parkinson. I don't know about this.
Because if I remember correctly, it's just a food for thought. It could be applicable, for example, to something related to the GABAergic transmission as well, for example. So, for instance, towards epilepsy treatment.
Okay. The fact is that target-ligand reductase, as I show you, they regulate the entire redox homeostasis inside cells. For sure, it may have some role also in Parkinson and Alzheimer's disease, but I don't know if it can be considered a target against this disease. For sure, it is a target against cancer and against glioma and brain cancers, but I don't know for possibly. I mean, I would say possibly, but I don't have the news.
Okay, thank you so much. Thank you.
Francesco, can you tell us how important it is to collaborate with the industry to get the results? So can you do it everything by yourself, or it's useful to have a collaboration since the very beginning?
Allora, Anna, the problem is that we are trying to find an industry to carry on this work, but at present we are not collaborating with any companies. We are trying to do that. Probably our patent will open some of these roads, but at present we don't collaborate with pharmaceutical company, mostly because I guess they are not so much interested in this kind of neglected diseases. So they are interested just only if you will translate this information to other diseases that can have a bigger market. And what we are trying to do with the people in the US is to try to sell in some way, our inhibitors for animal diseases. Because several similar parasites, it also several animals and cats, and this is an idea that we have in our mind.
Which could be a good idea, I think, because I mean they're a part of, of the game. Okay, any other questions from the audience? Anything you want to know? Also, in terms of, say, career development for young investigators, do you think that this field could really attract people? I have this impression because, as you know, the new drug discovery is running very fast. So how we can say something to the students to have as an option to go for this type of research?
Yeah, so I would say a couple of things about this. So the first thing is that the best research in this field is carried out north of Europe and the US. This is the first thing. But also in Italy, there are very good groups. And the other thing that I should say is that if you choose structural biology as a discipline of your career, you cannot expect to publish a lot of work. And this, because it is a very slow work. It takes years to solve a structure, both by X-ray and cryo-EM. So you cannot expect to publish several papers. Probably if you can think to place few papers in very good journal. This is right, but you cannot expect to publish a lot of papers. But for sure, in the future, structural biology, and in particular structure-based drug discovery, will be a crucial discipline. What I can tell also, another thing is that several big pharmaceutical companies, such as Merck in the US, they have their own cryo-EM microscope, and they are doing pharmaceutical research with cryo-EM. And consider also that in most of the drugs that are now on the market were discovered by the so-called blind approach. So, for example, praziquantel is one of these examples. I mean, it is not known the target of praziquantel. It is very effective. It was found by Bayer, I think in the 70s, working on drug active on the central nervous system. So what a pharmaceutical company did is try every drug they have against several diseases and select one that is the most effective without side effects. Now, every pharmaceutical company that would like to start to find a new drugs, the target must be known, the structure of the target must be known, and if you don't have this information, you have to get it, to get them to start a drug design study. And this will be crucial in the future and is crucial now. And the most recent example is the one that I showed to you about COVID-19 main protease.
Yeah. Oh, I understand. However, as an old scientist, I must say that, at least for my experience, first of all, this is for young people. Patenting is also part of the CBE. Now it is absolutely a positive point if you have patents. And in my personal experience, making pharmacological research and having patent doesn't prevent publications at all. So just a matter of timing, but you can publish, so it's not that the major issue. But as you say, if then maybe you publish less, but publish better, that could be good.
Absolutely.
Yeah. Okay, so I think that we are at the end of this webinar. I want just to inform you that the next webinars will also be interesting. The next one will be by Vincenzo Rizzi from the University of L'Aquila on physics of cycling, the point of view of an experiment. Then on the 25th of May, we will have Milena Vitale, a former student from the University of L'Aquila that is working now with Teleton. And it's about the career development. The title is How I Got Here from Bench Side to Rear Side. And on the 1st of June, we will have Ciro Menale from Naples. We don't have the title yet, but he will talk, I think, on something related to bone metabolism. So, and this is all. And thank you very much, Francesco. I think
that
your presentation has been really inspiring for us. And I hope to see you very soon.
Bye.
Thank you.
Thank you again.
Bye-bye.
Bye-bye. Bye, everybody.